Do I just start?
Please.
Okay. So what my project was the synthesis and characterization of inorganic fluorine tungsten disulfide composites. And first I'm going to talk about myself and my background.
Can you hear her in the back?
No?
I see no.
So first I'm going to talk a little bit about myself and my background, and then I'll go into my research and then lessons learned. So I graduated from North High in 2009, and then right after that summer I came into Hartnell College. And I came in wanting to be a social and behavioral science major. But then once I started working for Gear Up at Alisal, I was a tutor and mostly in biology classes, and that got me really interested in that. So I changed my mind, and I'm currently a biology major at CSUMB, and it's my first semester. So what my project plan at the Naval Postgraduate School was to create inorganic fluorine tungsten disulfide nanostructures. And what they are, they're like nanoparticles or nanostructures that are used as shock absorbers, like in helmets and armor. And the reason we want them spherical is, as you can see, like they have different layers. And as it gets impacted, only the top layer will break, and then the rest of the layers can be used as like shock absorbers. And we used a total of 2 methods. First was the atmospheric microwave plasma system, and what this does is it heats up the sample to about 100 degrees Celsius, and it speeds up the molecules faster since it goes through the plasma. And then the second method was the furnace, and in the furnace it heats up to about 600 degrees Celsius, and we used nitrogen and hydrogen disulfide to sulfidize it so the sheets would separate. And these are commercial products that we used in our project. And as you can see, the nanostructures all around, they're not really spherical. So what we did is we crushed them, and then we ran them through the plasma and the furnace. And the last step was to create the graphene, and we added the graphene to strengthen it and have it be more impact resistant. And first we added the acids to oxidize it. And once we got the thermally exfoliated graphene right here, as you can see, the sheets are separated farther apart, and it helped the spherical shapes go through easier. And these are the results we obtained through the scanning electron microscopy. And as you can see, we obtained more spherical shapes in the structures, and the plasma had greater results. And what I think I learned here at Hartnell and after transferring is that you guys should really appreciate like what Hartnell has to offer you and like the opportunities it has. And even though it might seem not as much right now, or like it's just whatever, it really helps in the long run, like who you know and who like you get to know, because they create different opportunities that can help you a lot in the future. And that's pretty much it. Any questions?
You're just entering CSUMB this semester?
Yeah, I just transferred this semester.
So when you say it helps you know, who do you
Allison at MPS, and then here Andy, and who else? My mentor Claudia, Joe Welch.
So is this research you're going to continue?
I'm thinking about it. I'm not sure yet though. Yeah.
I have a question.
Go.
So what are the applications of graphene?
Good question.
What do you mean?
Like what you're trying to make graphene with graphite in the infrastructure, and what are they use it for?
Well, what they wanted to do is like mix graphene with the tungsten disulfide. So the graphene is like layers, and then the tungsten disulfide would just be spherical shapes in between those layers. So they would be greater impact resistance, like in helmets and armor.
Say that again, last part.
Oh.
Impact resistance.
Impact resistant?
Impact resistant?
Yeah.
Armor?
And helmets.
Helmets and...
Some examples, there's more, but
So it would be like, and also like in bike helmets as well?
yeah, bike helmets, like the vest the cops wear and stuff.
Oh, that too?
Yeah.
So it's flexible, like bands, brakes?
Yeah, because there's like different layers. You see, that's like a nanoparticle, and you see how there's different layers, and if like it's impacted by any force, only the top layer will break, so you have like the rest of the layers as protection.
Is that electron microscope, is that ESG?
No, that was at MPS,
NPS?
the Naval Postgraduate School in Monterey.
Where would you find one, like where would you find a nanoparticle? And where would you find it?
What do you mean? Like where did
Where could I find that?
Well, are you asking?
Like in what?
Particles, are there nanoparticles found in nature?
Yeah, are they found in nature?
Or are these just man-made?
Or is it man?
Well, they're pretty much man-made. Yeah.
How do you make one of those?
Well, what we used was just commercial products, so they were already made for us. Like we bought it like on the internet over there.
Oh,
Do you,
okay.
Yeah.
do you buy the nanoparticles?
Yeah, they purchased them.
You guys don't make it though?
No, we don't make it.
Oh,
Like we just altered their like.
okay. You can make it better or just?
Yeah, cause let me see. You see, these are like commercial products. These were like the ones we bought, but they're not really spherical. So what we were trying to do is like crush them and then run them through the plasma or the furnace to heat it up, so they would be more spherical. Cause it's like better qualities and.
So which one's the plasma and which is the other one?
This one is the plasma, and this one heats it up higher, like up to 1,000 degrees Celsius. And this one is the furnace. It's kind of like an oven. It only heats it up to like 600 degrees Celsius.
And you don't have a finished product of what you're looking at?
Yeah,
Which one?
these two.
The one
They're
on the
like
left
a-
is the plasma?
Yeah,
That's the hot one, right? So with that one, which one is better?
we found out that the plasma was better since it heated up to like higher temperatures. We were able to obtain more spherical shapes and like in the long run.
To the bread, could you cut it with like a little knife?
No, it's like powder.
Or just powder?
Yeah.
And then when you do the powder, mix it in what?
Well, we would mix it with the graphene and then put it in either the furnace or the furnace, and then it would be like located right here, and we'd just like let it sit there for like 2 hours, and then we would take it out and then just test it like in the scanning electron microscopy.
Is it turned into cloth or rubber?
No,
What does it turn into?
just powder.
Oh, just powder?
Yeah, sometimes it would change color, but it's still like powder.
So after that process, you have to turn it into like a cloth, into different types of material?
Yeah, well, what our project was just to like create spherical shapes, but then there was another person doing another project, and they were creating the cloth, and then they would like successfully mix them together.
I'm familiar with the Teflon, the vest. You guys compare those at all? Like,
No.
As far as, no?
What's more effective?
Yeah, more effective.
No, we didn't compare.
That might be sort of the next.
Yes.
Are you planning to research this on the side by yourself?
No. Yeah, I just said I'm here.
So do you know of other institutions that are looking into this? So it's at the Naval Postgraduate School. Were there other groups that you collaborated with in other places?
No, just the Naval Postgraduate School. Yeah.
Do you want to talk about how CSUMB is a little bit, is different or similar to your experience at Hartnell?
Hartnell?
Yeah.
Well, I think it's pretty much the same. Just classes are a little bit more intense since like they're expecting more of you since like you're at the university. But the teachers are pretty much the same. Yeah.
So you talk to them like office hours and
They have office hours and everything.
Do you have tutors?
Yeah, like with the math courses they have over there, they have like 3 tutors. So like while the teacher's explaining, like if you have any questions, just like call the tutor and they'll come and help you, or like do the problems with you and help you understand.
Okay. Any other questions or comments? So let's take one more time. Alex, go ahead and start your talk. You ready?
Okay.
Yeah. Okay, so we got Alex Scala. He's going to talk a little bit about his path from starting at Santa Clara University, but eventually transferring to CSUMB and how he's close to finishing his degree in biology now. So he's going to speak a little bit about his experience doing some research at CSUMB and that trajectory of how he started in one place and ended up at CSUMB. So let's go ahead and welcome Alex.
So, I'm Alex, and I'm a senior at CSUMB. I'm majoring in biology with a concentration in the pre-med track. I don't necessarily want to be a doctor, but I like those classes. So, I'm going to talk to you today about undergraduate research opportunities at CSUMB. So, let's get right into it. So, before I start, I just wanted to give a little brief introduction about who I am and my background. So, I graduated high school back in 2009, and initially I went to Santa Clara University. And fantastic school, lots of help for you, but unfortunately it's a little bit on the pricey side. So unfortunately, due to some financial reasons, I ended up withdrawing from the university and coming here to Hartnell College. Now, I love Hartnell. I think all the stuff they have for you guys here is amazing. They have a ton of opportunities for you guys to do different internships, and the professors are very, very helpful. And so from here, I started thinking about, where do I want to transfer and where do I want to go? And the way I answered that question was, well, what do I want to do? I wanted to go off and one day become, some type of a researcher, maybe be a professor, that type of deal. So I wanted to get some experience in that and really decide, is that what I want to do? And so what I found out when I was researching the different schools is that CSUMB is very unique in that they have an entire program dedicated to getting you into internships and getting you experience in the real world of science, of doing research and being a part of a lab and getting these experiences. And so that weighed really heavily on my decision. I mean, I could have gone to a few different schools, but because they had this program, they were kind of what I leaned towards. And that was very, very important for me because honestly, when you're going to apply for graduate school, you want to know that there's two main reasons. You want to know for sure that's what you want to do. And secondly, you want to be a good applicant. You want to make yourself look good. So doing these internships, they really help you really solidify that this is what you want to do and you really enjoy this. And it just looks great because when you apply, you're like, oh, I have all these research skills. I've been in the lab for this long. And it looks really nice. So that being said, how do you get into these different research programs? And they have a research program here through Access, actually, which I strongly suggest you take advantage of, which is Access through, and it takes you to UC Santa Cruz, I believe. But anyways, so that brings me to UROC, which is the Undergraduate Research Opportunity Center at CSUMB. And the reason that I wanted to do this is because it's really, really hard to get into a program on your own. I mean, it's not impossible, and I don't want to deter you from that, but it can be very difficult. It can be very challenging and scary. Because I applied at UC Berkeley for a different, they call them REUs, Research Experiences for Undergraduates. And they sent me an email just saying straight up, we got over 300 applicants for 10 positions, and we're sorry, but we're not going to be able to accept you. And so it can be dismotivating. But fortunately, I went to CSUMB, and UROC got my application, and they started helping me and started figuring out how to make me a better applicant, how to make me more attractive to these different people. They helped me write my resume, write my applications, and just do things as best as I could so that I can look the best and get into these programs. Now, at the same time, the professors at CSUMB really helped me out. My professors, Henrik Kabak and Aparna Srinivasan, they were very, very involved in my academic life. They wanted to know what I wanted to do, where I wanted to go, why I was doing this. I wasn't just another person in the class. And so last summer, I was in Dr. Srinivasan's genetics class, and she approached me towards the end and said, hey, how would you like to work in my lab? Because she'd been talking about it the entire time, saying, hey, we have this lab here, we do this, this, and this. And she came up to me and asked me, how would I like to do it? And I said, sure, why not? And that right there is just really, really fantastic. That you can have someone just come and say, hey, come work in my lab. Like I just said, I just applied to Berkeley to work in their lab, and they told me, no, you're not even, we got 300 other people who want to come here. But I was able to get into this lab, fairly easily. And I know she's still looking for more people to be in her lab who are willing, motivated, and could use it. And so the name of the lab was the Molecular Ecology Lab there at CSUMB. And they're studying a few different lakes. First one is this one right here, which is Pinto Lake up in Watsonville. And the other one, I was trying to get a picture before class, but I couldn't. Is Loch Lomond. They're pretty well-known lakes. And what they're studying is something that you've probably all heard about if you've taken your first bio classes, which are CHABs, or cyanobacterial harmful algal blooms. And what that is, just of it is just, you have these cyanobacteria that live in these freshwater lakes, and when the nutrient levels get a little too high, they bloom. And you get all these guys popping up, and as a result, you get a lot of toxins sometimes put into the water. Particularly, this lab wants to look at a toxin called microcystis. And if you could see this picture a little bit better, these are probably another guy called phenazamanon. But you see there are tiny little clumps that form in the water, and that's microcystis. And they produce a toxin called microcystin. And it's actually encoded for in their genes. So these little cyanobacteria, they have it in their genes, and they may or may not be expressing them. So working in the lab was actually really fun. We get to go out to this lake, we get to ride on a boat and see. That's actually one of the stations right there where we sample at. It's a little buoy that monitors the weather. And we go out there and we have all this different equipment that takes depth samples for us. It monitors the conductivity and just everything that's going on in the water. And we take these surface samples it's a small, about 1 liter amber bottle to keep light from going in. And I was going to bring that too, but I didn't have it. But it's really fun, and it's really, really relaxing. Especially I work at Loch Lomond right now, and it's closed, but I still have access to the lake, so I get like the whole lake to myself. And it's really kind of neat. It's sort of eerie sometimes, but it's neat. And so working in there for a while, Aparna, my professor, asked me what I wanted to do as far as my own research idea. And at first I was kind of thrown back because it's sort of scary when someone comes up and tells you, come up with an idea, and to research on your own. And it was a little weird, but you would be surprised how many questions there are out there to be answered. And so one day I was just sitting doing something called cell counts, where we take a picture of the actual water, a sample, on a slide, and we look at how many different cyanobacteria are on there, how many colonies there are. And I was noticing there was this little critter who sort of looked like microcystis, but sort of didn't. And so I talked to the graduate student I work for, her name's Erin Stanfield, and I talked to my professor, and both of them were like, well, I don't know what it is. And so I said, well, how about that? Why don't I go ahead and find out what this guy is? And they said, that's fantastic. I started writing a proposal with her help, of course, to actually sequence this guy's genome, and not only that, run some PCR, which, for those of you who aren't familiar with it, is polymerase chain reaction. And what it is, we get these little primers, and we actually try to see if a certain piece of DNA is in there, and whether or not they can produce it. And of course, we're concerned with their ability to produce microcystins. And so that was my idea. Let's see if this guy makes toxins. If he makes it, what is he? And add it to the knowledge. And so this was another really key skill that I gained, was how to write a proposal. You know, because as a professor at a university, they're constantly writing grants, trying to get money, trying to convince people to believe in their projects. And it's very, very useful, because you talk to most of the scientists that I've talked to, people like you, people who want to become scientists, they don't really like writing, and they don't practice it. And so when they go to do these proposals, they kind of struggle through it. So actually being able to work with a partner who actually has she went to get another, was it a master's, I believe, in actual writing. So she was really helpful in helping me to figure out how to write, how to be a better writer and do well in these proposals. So in the matter of like a month, I'm just gaining all these skills and learning all about these labs, and it was just really, really fun, to be honest with you. It's so much work, but you're just having so much fun. You get to go to work and be happy. You know, it's a nice feeling. And so one of the things that's sort of key with all this research is, what does it mean in the long run? Because you can get caught up, you know, in these little tiny cyanobacteria and like, oh, what's this and oh, what's that? But you always want to kind of think about, well, what's the real importance of what I'm doing here? And so it's actually really important. Pinto Lake and Loch Lomond serve as a water supply, and people fish in those lakes for thousands of people, all of Watsonville and Santa Cruz. Santa Cruz is where Loch Lomond is at. And if these cyanobacterial blooms get to the point where they're really, really bad, microcystin has been known to cause liver failure in humans and mammals in general. So it's really, really important, because this is the water you're potentially drinking. You know, I'll get to your question at the end of the slide. And another thing is, besides the just general impact on the community, you guys have probably heard of model organisms, like, you know, Drosophila. I think yeast is another model organism. And they're just organisms that are really, really easy to work with. They're perfect for experiments, and scientists just love them. We do all our experiments on them. What you probably never heard of is a model lake. So we're lucky that we live here, because there's a lot of freshwater lakes around. Other areas of the US, and there's not so many. So there's not a whole lot of research being done on freshwater CHABs in lakes. So one of the key things about Pinto is it's really, really predictable. Through all the research that they've been doing on this lake, they've noticed a nice, you know, little up and down trend of when it's, like, you know, in October the bloom should be here, September the bloom should be there, and it's really, really predictable. So they want to actually use Pinto Lake as a model lake, so that when other people are studying freshwater CHABs, they can compare it to Pinto and see how their research stacks up. And then, of course, with just about any kind of research, there's just the general expansion of knowledge. As scientists, we want to know. We want to know how things work. We want to know why they work the way they work. And through my personal research, you know, we'll be able to know, okay, there's one more new taxa. It's a microcystin producer, and it's very common in freshwater lakes, hopefully. And so, more or less, coming here and going to CSUMB, I feel like I've gained more than I would have if I even just stayed at Santa Clara. It's just, there's so many opportunities for you out there, and you want to just set yourself up to have the best chance for yourself to accomplish your goals. So that's basically all I got. If you guys have any questions, I'd be more than happy to answer them. Go ahead.
Yeah, so how come you weren't able to come up with the money? You didn't have a scholarship?
At Santa Clara University?
Right.
It's about $42,000.
So how much did you...
So I didn't get anything. My family makes too much money to qualify for the Pell Grant. They're tiny microorganisms.
How do you spell it?
Cyanobacteria. It's...
And how do you find this bacteria? What makes this bacteria come alive?
What makes them come alive? Just same thing that makes you come alive.
And according...
No. They reproduce on their own. They just divide. The same way your cells just divide and you grow,
Yeah.
same way they grow.
Okay.
And so they're still alive. They're still their own little organism. I won't say animal. Their own little organism. And so they produce toxins. The same way, you'll defend yourself if like a dog attacks you. They produce these toxins to keep things from bugging them. So the problem is when like, let's say, there's actually some strawberry fields right around here.
Does that affect strawberries?
These guys, I want to say no, but you never know.
Can you cure them? Can you cure the water?
Right. So like these guys will bring, like I said, there would be a strawberry field right here, and it runs nutrients into here. And what happens is these guys take up those nutrients and they bloom beyond what you would normally see. And so that's where it becomes dangerous, because then there's so many of them producing all of these toxins, the lake turns really, really green like this, and it's bad for you. Now you can fix this lake, and there's another project working there that's actually trying to get rid of all these cyanobacteria. And what they're doing is they're using this chemical called alum to get rid of, I don't know the details of their project and how the alum actually works. I know it's a problem because it sinks to the bottom and it covers the sediment, but they're working on trying to find a nice balance. And Loch Lomond is a treated lake. I should have mentioned that. Loch Lomond is actually a treated lake chemically. And what we do is we compare this lake, this untreated lake, to Loch Lomond, which is a treated lake, and see, when the Loch Lomond bloom tries to happen and then they treat it, we examine it and we see how it's working, see just to kind of compare the two and see if it's being effective.
My request, is Europe available to Hartnell students too, as in like the CSUMB question? Like if you want to transfer there, can you go before and like be like?
I don't know about that, but the other thing that I forgot to mention is that when I first started in the lab, I worked with this student named Cindy Cho, and she was a high school senior. And all she did was she emailed Aperna. She emailed a ton of teachers, but she emailed Aperna and said, hey, is there any chance I could work in your lab? And she just said, yeah. So sometimes it's as easy as that. You send someone an email and they say, sure, come and work. So if you guys want to all send Aperna an email, you can look her. I'm not gonna put her email up there because this way I'm not responsible. But you can go ahead and look on CSUMB's website. You can find Aperna's email and you can email her and see if she'll let you work in her lab. What's up?
Yeah. So when you compare the two lakes and how the chemically treated one differed from the other one, you have already had samples happen.
Oh yeah, these samples they've been.
What was the difference?
I personally haven't looked at these samples. I'm barely starting doing PCR on this guy because I should have mentioned this too. I've only been working in this lab for about 3 months. And so in that 3 months, I've learned how to take samples. I'm barely starting on doing PCR. I've done PCR before, but doing all the actual PCR steps. I have learned how to use a depth sampler. It's called a HydroLab to figure out the conductivity that's going on in there, the level of penetration of light. We do a lot of different filtrations to collect different chlorophyll A samples, toxin samples. There's a bunch of that type of deal. Collection, filtration, basic PCR. That's what I've done so far. I haven't actually done the big product of examining both, but that's actually the next thing that Aperna wants me to work on.
How old are you?
21.
All right. You ready for another question?
You guys,
Yep.
Is this the research connected to the marine biology department at CSUMB?
to the marine biology department at CSUMB. Maybe. I don't think so. It's mainly just genetics and molecular ecology.
Molecular ecology?
Yeah.
So actually, speaking of what makes you go, I was going to say, we're going to have pizza. So if you want to ask more questions, you can ask as many questions of this guy and Marissa as you like. We're going to head over for some free pizza at Mountain Mike. Are you familiar with Mountain Mike? Or the close one? What I want to do is, and actually, Jose, are you going to be there? Yeah, I'm going to be there. So I want to invite you to come to pizza. let's go ahead and thank Alex. I set some cards around for our first set of speakers, so that'd be great if I could get those cards back. And it would be also great if, yes. Oh, okay, so if you need to sign them, I'll put them up front, or you can get them from Kevin.
I thought you laughed.
Because, well, somebody is paying for this pizza, and